Good afternoon, everyone. My name is Christine Ryan, and I am a fourth-year graduate student in the Miami project in the lab of Dr. Jay Lee. And our lab studies spinal cord injury as a wound healing process. And so we look a lot at some of the non-neuronal cells at the injury site, including immune cells and glial cells. So I'm excited to share my research with you this afternoon. So as you guys are all aware, after the injury to the central nervous system, mechanical damage to the blood-brain barrier in the tissue itself induces a large influx of infiltrating immune cells, including macrophages. Macrophages are the professional phagocytes of the body, and they participate in a number of functions after injury, including inflammation through the release of cytokines and various other signaling factors. They also directly contribute to angiogenesis, gliosis, and fibrosis occurring in the tissue after injury. As far as the timeline, immediately after injury, there tends to be a local glial response. And then by 1 to 3 days, you start to see the influx of these cells, notably macrophages, and this peaks at around 7 days. But as you can see, they remain a sustained population in the cord from weeks to months after injury. So why are we interested in macrophages? Because macrophages are so essential to the wound healing process in the periphery, as well as the central nervous system, and are so crucial in orchestrating the inflammatory response after injury. A lot of study has gone into kind of elucidating their role in injury, and there is some controversy about exactly what they're doing at the injury site. So for this reason, it's a focus of our lab's research. So in order to gain an unbiased glimpse into what these cells are doing after spinal cord injury, previous work from the lab shows that we isolated macrophages from the 3-day post-injury site and the 7-day post-injury site, and basically sequenced these cells, and basically to get a snapshot into their transcriptional profile and hopefully to glean some insight into their function at that time. And so when we compared the transcriptome of macrophages at the 3-day versus 7-day post-injury mark, we were actually quite surprised to see that at 7 days, the gene ontology biological processes that were enriched in this gene set basically were mostly related to lipid processing and lipid metabolism. This was not really expected for us, but even when we compared the transcriptome of these 7-day post-injury macrophages obtained from our study to that of the transcriptome of other publicly available data sets, we found that they had the greatest transcriptional similarity to that of foam cells. And we'll get a little more into what foam cells are. So taking a step back, after injury, in the central nervous system at rest, in a normal, healthy, functioning nervous system, we have our neurons that are ensheathed when this fatty substance known as myelin, which is produced by glial cells known as oligodendrocytes. So, however, after injury, you get damage to these neurons and to their myelin sheath. And so this results in the generation of a large amount of lipid and cellular debris that must be cleared in order for healing to occur. And so when the macrophages come in, they begin to eat up this lipid debris. But lipids are notoriously difficult to process metabolically. And basically, these cells become overwhelmed with this task and become filled with lipid droplets, as you can see here. And so this is what gives them their quote unquote foamy phenotype. So foam cells have been described in a variety of other diseases, namely atherosclerosis or heart disease, where they operate by a similar mechanism. Basically, in the case of atherosclerosis, there's a buildup of cholesterol in the arteries, and macrophages are recruited by local inflammatory cues to come in and clear this debris. And very similar to spinal cord injury, they begin to eat up these lipids, and they basically, as I mentioned, become overwhelmed with this process. And this results in a number of downstream consequences that contribute to cellular dysfunction. Includes impairments in lysosomal processing, the formation of these large lipid droplets, the induction of pro-inflammatory signaling and the release of cytokines and chemokines. So this kind of creates a feed-forward positive feedback loop, worsening inflammation and often outcomes. And cell can become so stressed that they even undergo apoptosis. So work from another lab at Florida State has also been interested in foamy macrophages after spinal cord injury. So I took this image from a paper of theirs because I think it nicely shows the kind of timeline during which we can see foamy macrophages after spinal cord injury. So up here we have the normal cord, uninjured cord. And at 3 days post-injury, remember when the macrophages are just starting to come in, you don't really see the appearance of any of the lipid debris. However, by 1 week, we start to see this signal. This is a lipid dye known as oil red O. So you can see the red mark is the lipids, neutral lipid droplets are accumulating. And so by 1 week, we start to see the formation of these droplets. The macrophages have been there for days now and have been clearing away debris. And basically, this just worsens over time. By 2 weeks, you see even greater extent of this signal. And by 4 weeks, the injury has kind of condensed into this lesion core, and it's basically filled with these huge round macrophages that have been basically engorged with these lipid droplets. So they also wanted to see kind of functionally what these foam cells are doing. And so they took cultured bone marrow derived macrophages and treated them with purified myelin, and basically measured some of the cytokine response of these cells. And with treatment of myelin debris for 24 hours, they found elevated levels of many pro-inflammatory cytokines, including IL-6, IL-1 beta, TNF, and many more. So with this kind of as the background for my project, I wanted to kind of give you the big picture into what I've been studying. So as I mentioned, we've noticed that at the injury site, there's a large amount of lipid debris. That uptake of this results in the accumulation of lipids inside the cell. However, a big assumption in the field is that myelin is basically the main source of this lipid debris that's driving this process. Given that myelin is such an important lipid in the central nervous system, it makes sense why that would be an assumption. People even have gone so far as to model this in vitro by just using purified myelin, indicating that really that's the only important constituent of the injury site that's driving foam cell formation. However, this really hasn't been directly tested. So first idea we wanted to tackle is really clarifying what is the role of myelin in driving this process. So we wanted to, again, look at some of the intrinsic changes that are occurring in the foam cells due to this lipid accumulation. There have been reports of cellular dysfunction. And so we wanted to see metabolically what is occurring within these cells that as a result of this lipid loading. And lastly, as I mentioned, macrophages, specifically foam cells, have been long associated with inflammation. And so because our lab is so interested in inflammation after spinal cord injury, we really also wanted to characterize their inflammatory response to the injury site. So as I mentioned, first things first, we wanted to determine the contribution of myelin to foamy macrophage formation after injury, with our hypothesis being that increasing myelin content will suppress foam cell formation. So in order to answer this question, we utilized a pretty unique mouse model where a mutation in myelin basic protein renders these mice basically devoid of central nervous myelin. So little image here, they're also called shiver mice because, as I mentioned, they don't have any myelin, so they do have impaired neuronal conduction in their central nervous system, in an uninjured state. But this gives us a really useful tool in order to answer this question. When you take away myelin from the central nervous system, do foam cells form? So this is just also right here, some histology taken from the shiver mice. At the top, we have the Luxol fast blue stain, which stains myelin. So you can see in the wild type animal, they have a normal robust stain. And in the shiver cord, completely devoid of any signal. And again, this top panel is the wild type mouse. We see a longitudinal section of the cord, and you can see intense staining of myelin basic protein, or MBP, in the white matter tracts of the wild type cord. But in the shiver mouse, this is completely gone. So to really directly test the role of myelin in foam cell formation, we cultured bone marrow derived macrophages. As I mentioned, the kind of pre-existing model for modeling foam cell formation in vitro was to basically purify myelin from the cord, treat macrophages with this, and generate foam cells in a dish. However, we thought it was better to contain the entire spinal cord homogenate. That way, we're accounting for the entirety of the lipid constituents that are, in the spinal cord. So, to directly test the role of myelin in foam cell formation, we treated these cultured macrophages with spinal cord homogenate from MBP het cord, which contains a normal amount of myelin, and then the MBP knockout cord, which again contains no myelin, but there are other sources of lipids there, such as cellular debris from dead and dying cells, and other sources. So when we did this, it's pretty interesting. As is expected, well, first of all, foam cells formed, which was what we expected. We have the appearance of these lipid droplets here in green. This is the lipid dye we use in vitro. The cell mask, this is an outline of the cell body in red, and then the nuclei in blue. But this top panel here shows the cells treated with the MBP het cord, which is again the normal cord containing a normal amount of myelin. And as you increase the percentage of the homogenate that we treated the cells with, you see a dose-dependent increase in the number of lipid droplets. And at 1%, they're basically filled with these tiny lipid droplets. However, when you treat with MBP knockout cord, which again does not contain any myelin, you see the appearance of lipid droplets, although it is to a lesser degree when you compare to the same percentage of the MBP het cord. However, lipid droplets are still forming, which is not really what we expected, to have this robust response to the myelin-less homogenate. So again, here we have quantification for the spot intensity of the lipid droplets in these cells. And as you can see, it's about for the cells treated with the knockout homogenate, there is a significant decrease in the lipid droplet fluorescence, but there, yeah.
I'm sorry, but on my screen, I now have your title slide. I don't know if that happened to anyone else.
Me too.
Yeah, me too.
Yeah.
Oh, thank you so much. All right, continue on. Alrighty, are we on the lipid droplets slide, or is it still this title slide?
Titles. Okay, now we're talking.
Okay. Let me get my pointer. Alrighty. Is it still the lipid droplet slide? The at and knockout?
Yes.
Now we lost Christine.
Yes, so it's we have the right screen now, just don't have Christine. But on the plus side, we have a picture of Jay.
See what you did, John? You scared her away.
Hi, sorry about that, everyone. Some technical difficulties. Okay, so sorry about that again. So take home from this slide, again, when we treat these cells with the knockout cord, we still get the appearance of lipid droplets, indicating that myelin is not the only source of lipids driving foam cell formation. So kind of as an extension of this experiment, we subjected the MVP het and the MVP knockout mice to conducive spinal cord injury. And here we have our macrophages and microglia labeled in green with CB11B stain. And then here for in vivo stain, we used oil redo to stain our neutral lipid droplets. And so at 7 days post-injury, this is the het cord, which is again contains a normal amount of myelin. You see the appearance of these lipid droplet-filled macrophages. However, in the MVP knockout cord, there is a reduction in the intensity and the area coverage of this oil redo signal. But again, as I mentioned, there is still foam cells forming, again, in these mice that have no myelin, which is indicating to us that, again, myelin is not the only source of lipids, and that the source of the contribution of just general cellular debris is also important for this. This phenotype was also seen at 4 weeks post-injury. Here again, the lesion kind of condenses into this lesion core, and you see the formation of these large round lipid droplet-filled cells. And while this is again smaller in the NBP knockout mice, foam cells are still forming and basically recapitulating what we saw both at 7 days post-injury as well as using our kind of new in vitro assay. We also wanted to look at other aspects of the histology at the spinal cord injury site. So we also measured the lesion size, which is the GFAP negative lesion area, with the GFAP positive being the spared neural tissue, where you can see the presence of the neural stain Myelin. So when we compared that between the NBP het and the NBP knockout mice, we also saw a reduction in the GFAP negative lesion size. Again, when we measured this at 28 days post-injury, a more chronic time point, we also saw a reduction in the size and area of the GFAP negative lesion. So for this first part, our main take-home points is that, again, myelin is the source of lipids, but is not the only nor most important source. Foam cells still form in the absence of myelin, and so we should take this into account when we model this in vitro. So this decrease in lipid droplet formation also resulted in a decrease in GFAP negative lesion size, and it's suggesting that the lipid droplets in the foamy macrophages are important in driving other aspects of the spinal cord injury pathology. So next, we wanted to address some of the intrinsic changes occurring in foamy macrophages after injury. So namely their metabolic state. It's been known that lipid droplet accumulation in other cell types can result in a number of features of cellular dysfunction, including metabolic dysfunction. So to test this, we used a pretty cool technology known as seahorse adjuvant technology. And basically, what you're able to do with this machine is you have cells in culture, and in real time, you can measure their oxygen consumption rate. So you subject the cells to the injection of a series of different compounds that have a known effect on the mitochondrial electron transport chain. And then based on that, the response to these compounds, you can calculate a variety of parameters that kind of describe the cell's energetic and metabolic state. So first, you have the injection of oligomycin, which is an inhibitor of ATP synthase, which is part of, number 5 in the electron transport chain. And this results in a reduction in the oxygen consumption rate. And basically, this reduction corresponds to the ATP reduction that is occurring in the cell. Any remaining oxygen consumption in this after ATP synthase has been inhibited is due to proton leak across the membrane. So there's a number of reasons for proton leak, but one of the most important is when the mitochondrial is what's called uncoupled, or leakiness across the membrane due to a number of reasons causes this kind of pathological state where the mitochondria is not functioning properly. So after the injection of oligomycin, you have the injection of FCCP, which is basically an uncoupler. It completely disrupts this proton gradient, which is normally what's kind of dictating the rate of oxygen consumption. So by doing this, you drive the cell's oxygen consumption rate to a theoretical maximum. And then based on this, you can calculate the cell's maximum respiration and the spare capacity, which is the ability of the cell to go above its basal respiratory rate to meet its energetic demands. This is another important kind of measure of mitochondrial health and function. assuming that usually around 50% spare capacity would be normal above basal respiration rate. Lastly, you inject rotinoin and antimyosin, which basically shuts down the remaining features of the electron transport chain. And so the entire electron transport chain is shut down, and any remaining oxygen consumption is due to non-mitochondrial sources. So this is the result comparing our media-treated macrophages with our foamy macrophages. I kind of separated them out into these two different graphs. We'll kind of go through each of the parameters as I mentioned, but kind of at first glance, you can really see that these foamy macrophages kind of have is much higher, although much higher levels of mitochondrial uncoupling. So that includes increased proton leak, seen here in purple, as well as reduction in spare respiratory capacity, seen in dark green. So this is the breakdown of the various parameters I mentioned with our media versus inhibited cells. There's an increase in basal respiration, that increase indicating that they're in an increased energetic demand state. This is also reflected by increase in ATP. However, when you measure the ratio of this basal respiration to ATP production, which is known as the coupling efficiency, i.e. how efficient is the cell at coupling respiration to ATP production, you see a significant decrease. Again, this is kind of another indicator that there's some mitochondrial dysfunction occurring in the foamy macrophages. And again, just highlighting what I showed you in the other slide was this decrease in spare respiratory capacity in the cells and this increase in proton leak across the mitochondrial membrane in the foamy macrophages. So next, based on our seahorse data, which is showing elevated oxidative state, as well as these hallmarks of mitochondrial uncoupling, kind of the first thing placed my mind went was reactive oxygen species, which can occur when basically your electron transport system is not working as it's supposed to, but you still have this very high energetic demand. So this results in the imperfect transfer of electrons through this chain and can cause the increased production of ROS, or reactive oxygen species. So to test this, we have a reactive oxygen species dye known as mitosox, and you can see it in red. Basically, you treat the cell with it and it reacts with the ROS inside the cell to create a signal. So for a positive control, we used hydrogen peroxide treatment to induce oxidative stress in the cells. And you can see a robust increase in the number of mitosox positive cells. And when you compare media versus homogeneous treated cells, you do indeed see an increase in the number of mitosox positive cells. So the conclusions for this aspect of my project is first and foremost that mitochondrial uncoupling occurs in the foamy macrophages. This includes increased proton leak, decreased spare capacity, and decreased coupling efficiency. Also, there's an increase in reactive oxygen species production. This can have many downstream consequences in the tissue and has been shown to be harmful to the surviving cells at the injury site. So this is kind of our first demonstration that foam cells are actively participating in the pathology of the injury site, as I mentioned, with the reactive oxygen species being known to being harmful to the cellular milieu of the injury site. So next on our list, we wanted to more robustly characterize the inflammatory signature of these foamy macrophages after injury. Our hypothesis being that they are indeed important drivers of inflammation after injury. So to do this, we used a cytokine array where we could basically do an unbiased screen of 111 different cytokines, and basically they come adsorbed to this nitrous cellulose membrane, They're in technical duplicates. That's why you see your dots in kind of pairs of circles. Basically, you take cell lysate or cell media and treat these membranes with that lysated media, and whatever is in that lysate will react with the membrane. And after you probe and visualize, you'll get basically a snapshot into the cytokine signature of your cell type. So for this experiment, we first compared media treated or just regular macrophages with that cell lysate of foamy macrophages that had been treated with homogenate. So when we compared the cytokine array of these two cells, we found a number of hits. There's definitely a greater number of cytokines upregulated in the foamy macrophages, but the most notable ones included CCL6, IL-1 alpha, shown here, GDF, CXCL16, and SPP1. So this is just a quantification for some of the most significant top hits. I also have taken from, so this is 4 biological replicates, 8 different membranes. So you take 1 membrane per condition, per biological replicate. And lined up, you can really see that there's a large increase in a number of these cytokines, as I mentioned, including IL-1 alpha, CCL6, SPP1, and more. So next comparison we made is we took lysate from foam cells generated from MBP het homogenate, which contains myelin, and cell lysate taken from cells treated with MBP knockout homogenate. And it was interesting to us that we didn't see a huge number of differences, but of the ones that were there, probably the most noticeable was this large decrease in IL-1 alpha in the knockout homogenate, which suggested that potentially we have a myelin-specific cytokine. We also saw an increase in MCSF, which was consistent with what we saw in our media-treated cells, which was higher. There was a decrease in the MCSF production in foamy macrophages. This is the only cytokine really in this study that showed decrease in foamy macrophages. So it was interesting that this was also higher in the knockout homogenate treated cells. So to see if this IL-1 alpha was purely myelin specific or just due to differences in lipid content, we found the percentage of MBP het homogenate that resulted in lipid droplets of equal fluorescence of that of MBP knockout homogenate. We found it was about a 5-fold difference. If we treat with 0.1% MBP het homogenate or 0.5% MBP knockout homogenate, you get cells with lipid droplets that are roughly equivalent. So this is qPCR of the IL-1 alpha. And you can see that although there's a dose-dependent increase in the production of IL-1 alpha, again at 0.5%, almost 60 to 70-fold increase, at this 0.1% het and 0.5% knockout, they are roughly similar, which is suggesting to us that this may actually just be more due to lipid content. We also performed this using just purified lipids from the spinal cord homogenate. And it was interesting. There was quite a bit of overlap and some notable similarities between the two, but there were also some different targets that came up, including SERPIN-E1, HGF, C1q, and cystatin. But some of the ones that were consistent with our foam cell assay included DL6, osteopontin, IL-1 alpha, and GDF15. So given that we now had this robust cytokine profile of our foamy macrophages, so this figure in cyan or aqua shows the full list of significantly upregulated or significantly changed cytokines in the media versus homogenate treated cells. So again, this is just the non-foamy and foamy comparison. We have our non-foamy cells here on the left and our foamy macrophages on the right. So given these cytokines, we subjected them to some pathway analysis to try to see what type of protein signaling networks may be underlying this cytokine response. And we got a hit of a number of pathways, including age-RAGE, the JAK-STAC pathway, and the PI3 kinase AKT signaling pathway. So to see if any of these actually are important in participating in this foam cell response, we got inhibitors for each of these pathways. We have the age-RAGE pathway inhibitor in green, the JAK-STAT pathway inhibitor in orange, and then TORN2, which is an mTOR inhibitor, which is a component of the PI3 AKT signaling axis. So we treated our foam cells with this pathway, or with these inhibitors. We did an hour pre-treatment and then co-treatment with the homogenate and the inhibitors, and compared it to our vehicle-treated foam cells. And as you can see, the TORN-treated cells, this is a 1 micromolar dose and a 10 micromolar dose here on the bottom, resulted in a robust reduction in the lipid droplets. This is quantified over here. There were reductions with the other two inhibitors, but they were nowhere nearly as strong as the TORN2 response. So for that reason, we decided to pursue TORN. So this is just verifying our results of our mini screen, where you see, again, this large reduction in the number of lipid droplets in the foam cells treated with the TORN2 compound. Also interesting to note here that does seem to be an alteration in the distribution of lipid droplet, in addition to both the decrease in lipid fluorescence and lipid droplet number. So next, we wanted to see if treatment with TORN2 at all altered other aspects of the cellular pathology observed due to homogeneous treatment. So we used our ROS-specific dye, the mitosox dye as well. Here we have our media-treated cells and our positive H2O2 control. And at our bottom, we have our 2 foamy macrophages, either treated with vehicle, so just our normal foam cells, or treated with TORN2. So you can see that this is the gray image is just a bright field image. You can see that, again, we see the same kind of reduction in the lipid droplet number, as well as distribution in these foamy macrophages treated with TORN2. And this results in a decrease in the number of ROS-specific cells. In fact, it actually equals levels similar to that seen in just the media-treated cells. It's also interesting to note that TORN2 had no effect in reducing the production of reactive oxygen species in our positive control or H2O2 cells, and seems to be selective for lipid-induced reactive oxygen species production, as evidenced by this decrease in the mitosox cells in the foamy cells, but not in our hydrogen peroxide controls. So just take-home picture of what I shared with you today. We discussed the role of myelin in this process, and using the MVP mice, we showed that myelin is not the only source of lipids driving foam cell formation in vivo, and thus we created a modified in vitro assay that, instead of using purified myelin, uses the entire spinal cord homogenate to account for all sources of lipids that the cells would be exposed to in the normal injury site. We also looked at some of the downstream consequences of this lipid accumulation, one of that being the metabolic dysfunction occurring as a result of this lipid loading. And we showed that there is mitochondrial uncoupling occurring using adjuvant seahorse technology, as well as an increase in reactive oxygen species production. Lastly, we also characterized the inflammatory profile of these foamy macrophages and identified several different cytokines that are selectively upregulated in foamy macrophages. One of them being IL-1 alpha. So we are doing some follow-up studies currently targeting both IL-1 alpha as well as the IL-1 alpha receptor at the injury site. So stay tuned for some more in vivo data. We also identified a target, mTOR, and treatment with Torin-2 also looks to be a promising target. So again, we are starting to look into some in vivo studies targeting macrophages after injury. And so with that, I would like to thank you all for being here. I would like to first and foremost thank my mentor, Jay Lee, and the rest of the Lee Lab, including Susanna, Lindsay, James, Yadira, and Shafayat, and Stephanie, our former grad student. Specifically, James and Shafayat helped with some recent in vivo processing, so special shout out to them. The neuroscience graduate program, Dr. Atkins and Maya Pono are both great support systems. And my committee, Dr. Abigail Hackham, my chair, Dr. Roberto Barambilla, Dr. Hassan Al-Ali, and Dr. Armando Mendez. I would also like to thank the Marias Lab for so generously letting me use their seahorse machine. And then also to Yanshi, Dr. Al-Ali, and the Fornoni Lab for assistance with opera imaging. So all those pretty pictures of the foam cells were definitely a collaborative effort. And lastly, thanks to Adnan Mukhtiar and Dr. Constantine Levy for help with the cytokine array and other protein processing. And of course, our funding sources. Okay, thank you. Any questions?
I have a question, Christine. This is John.
Sure.
I want to ask a little bit about your Torin-2 results.
Mm-hmm.
I think Dr. Al-Ali is on your committee. I know that you're acutely aware of dose response issues.
Yes.
So I wanted to ask how you chose the concentration that you use and what you think it's doing at that concentration.
That is a very good question, very valid. So this has come up. Dr. Ali has brought this up. So we did test other concentrations, even lower molar, 100 nanomolar, 1 micromolar, and 10 micromolar. I know 10 micromolar is higher than typically used in like a lot of cell culture studies, and certainly above the IC50 of Torin-2. However, because we're adding this compound alongside that homogenate, we figure you're losing, I mean, there's certainly proteins and other, components of the homogenate that are likely interfering with the pharmacology of Torin. And so I suspect that's part of the reason why we had to use kind of a higher dose than normal. But still, there is the concern that there may be some off-target effects at this higher concentration. We haven't seen a decrease or a large decrease in cell viability, so at least we know it's not just toxic effects of the compound. But we are currently doing siRNA studies to knock down mTOR and basically try and recapitulate what we saw. Unfortunately, macrophages, specifically primary macrophages, are notoriously difficult to transfect. So we actually will be, I'm troubleshooting right now to get consistent knockdown of the mTOR in my macrophages. And hopefully that will provide some clarity on whether is it mTOR specifically or some off-target kinases that would be responsible for this effect.
Thanks. So that it may do that. I guess my suggestion would be, just to clarify, the known primary activity of taurine-2 at low or reasonable concentrations is to inhibit mTOR
Mm-hmm.
in an ATP competitive manner. So it
Mm-hmm.
inhibits both mTOR-C1 and mTOR-C2.
Mm-hmm.
And in Hassan's experiments, for example, it does both completely, 100%, at a concentration of 100 nanomolar. So I guess the hypothesis would be, with your presence of all your lipids and whatnot, that wouldn't be the case in your culture system. But that's a testable hypothesis. You could directly ask whether mTOR activity is in fact inhibited and at what concentrations. That would be my suggestion, just a suggestion.
I see. So you mean doing an actual readout of mTOR activity to confirm
Right.
that is.
Which can be done. It's not as hard as it sounds, because you can look at a mTOR specific phosphosite on other proteins and measure phosphoproteins with a Western blot. And that would give you insight into whether your hypothesis about interference is correct.
Christine, you're muted.
There we go. That is a great suggestion, actually, and would definitely maybe not help us with off-target effects, but would at least confirm that mTOR is being.
Yeah,
actually,
I just mean if you know when mTOR is being inhibited,
yeah,
you know whether you're getting off-target effects if you have to go higher than that. That's the logic.
I see.
Before I leave, I should say that I thought it was an excellent talk, by the way. I forgot to say that.
Oh, well, thank you.
Christine, you're muted again.
I know I'm waiting for, there's some background noise, so I just while we were waiting for someone. If there are any other questions?
We also want to thank Hassan for his tremendous help with the assay.
Mm-hmm. He has been a huge help and asset to my committee for sure. He is the assay development king, so definitely.
Christine, I have a question if nobody else has one. You briefly mentioned purified lipids, testing purified lipids. Can you tell us why you did that experiment?
Sure. So we obviously the spinal cord homogenate has more than just lipids in the present in that kind of milieu. So we wanted to kind of try and see if we could separate the contribution of other factors, namely the kind of damps and PAMPs, the damage-associated molecular patterns, and other sources in the homogenate that could be, responsible, at least in part, for, driving some of the, cytokine production. So it was kind of like a control experiment and also kind of proof of concept. But there are some caveats with that experiment because, treating with purified lipids is different than treating with the homogenate. Not only a concentration issue, when you look at the foam cells, maybe it would be useful moving forward to include a picture of foam cells that were generated from the purified lipids, because they're definitely not as foamy as the ones treated with the homogenate. So you get a much more robust phagocytic response when you treat with the bulk homogenate, likely due to the presence of these PAMPs and DAMPs that are going to stimulate phagocytosis. So partly, I don't know if they're not foamy enough if you need to treat with a higher concentration of purified lipids, but the concentration I chose was based on the recommendation of Dr. Mendez, who has done a lot of ox-LDL and acetylated LDL treatment of macrophages. So that was kind of our motivation for distinguishing between the homogenate and the purified lipid. But it does have its caveats on its own, but yeah.
And I have another question, but I'd like to give anyone else who has a question a chance. I guess not. If,
All you.
yeah, if let's see, I don't think Kevin's here anymore, but I think if Kevin was here, he may have asked, and maybe he wanted to ask, you know, how are you going to inhibit mTOR without affecting some beneficial things like axonal growth? So I guess first of all, how do you think? So I mean, obviously, taurine. We think that taurine-2, or you think that taurine-2 is working through mTOR, and that's the molecular target. But how about the biology? I mean, what do you think? What about the foamy macrophage biology do you think taurine-2 is affecting?
Well, I would say mTOR is like a canonical like energy sensor. So I think that it's definitely would make sense to me that it has some role in sensing the abundance of lipid droplets and, you know, other kind of metabolites that the foam cells are producing or exposed to. So I think there's some type of energy sensing that's going on. And maybe by inhibiting mTOR, you are kind of dampening the response to the lipids and kind of capping the response, so to speak. So just limiting the amount of lipids that they're able to process. So it's kind of, I guess, for your first question, like biologically speaking, what is the mTOR doing in the foamy macrophages? And to your other point about mTOR, overexpression of mTOR has been shown to enhance regeneration of neurons. So I do understand the concern of, you know, maybe using a systemic mTOR inhibitor could have, you know, potentially negative effects on axon regeneration. There are ways to target macrophages specifically in vivo, and has been used in the clinic. So not just, you know, a mouse model or a knockout. You know, you can load taurine in liposomes, and that will specifically target macrophages because liposomes are preferentially taken up by macrophages, the phagocytes of the body. But I guess also, even if we didn't have liposomes and we just treated systemically with taurine, I mean, there could be potentially some effect on the neurons, but at the same time, there's the argument that we haven't even been able to get enough robust growth to actually, you know, enhance function after injury. So, you know, potentially, if the mTOR inhibitor has a greater effect on the foam cells, and the foam cells are, you know, contributing to inhibition of regeneration, whether through fibrosis or worsening inflammation, then it might kind of, the pros might outweigh the cons.
Okay.
All righty. I think that's it, but thank you everyone. And if you have any other questions, feel free to email me. Thanks so much.